I'll get started. So my name is Mark Chaisson. I'm an assistant professor at USC. It's my first year there, and I've got 100% of my lab is here. So I'm hoping everyone else can keep that record up for their labs going into the future. 100% of my non-postdoc lab. I have one lonely postdoc sitting downtown right now. So today I'm going to talk about detecting structural variation with single molecule sequencing. And it's a bit of a tutorial, a bit of a survey, and a little bit of background methods all kind of rolled into one. Are we doing organic pointing, or do we have a laser pointer?
We have a laser pointer with low battery.
Low battery, okay. So I'll generally kind of, okay, well this works. And I think we can use the Google pointer. Okay, that's better. So when I tend to think about detecting variation, I think of all the different scales of human variation. And when you think about different scales of human variation, you can also just sort of think about how the genome is structured on different scales, all the way from kind of small scale structure in terms of just coiling, and then large chromosomal structure. And the variance that we'd like to detect when we're doing disease studies is essentially all variation. So there's a lot of studies that do, say, SNP association or looking for large-scale insertions and deletions, but what we'd like to have is profiles of every individual with every base pair of variation resolved. So at the smallest scale, there's simply SNP variation. This has been detected for quite some time using microarrays and high-throughput sequencing. There's been what's called structural variation. These are greater insertions and deletions and inversions that are greater than 50 base pairs. It's a functionally defined cutoff. And some examples of these are mobile element insertions, such as ALU or L1 elements. And this has largely been detected by high-throughput sequencing, at least on the very small scale. So for example, microarrays can detect large-scale variation, but not necessarily 50 base pair variation. And then on the very largest end of the scale of variation is what I call sort of chromosomal scale. So this is large-scale, multi-megabase copy number gains and losses. And this is actually something that can be detected with many different techniques, all the way from cytogenetics through to high-throughput sequencing. Most recently, this type of variation has been detected most commonly using read depth-based methods in high-throughput sequencing. So even at the smallest scale, the most simple type of variation, such as SNPs, there's disagreement between the different callers. So you would hope that this problem gets resolved well enough that any given method that is applied to a data set will essentially give the same result. And any difference between the results is some sort of measurement of the amount of uncertainty that we have in detecting changes. So what's shown here is a recent study by Hung Lee, where he was using a pseudo-diploid study where he took two haploid cell lines and sequenced reads between the two of them and used that to estimate false discovery rates, and showed that, well, it's fairly low. You're measuring false discovery in terms of parts per million. There's still a measurable difference. And this becomes important when you're looking for very slight variations, such as Denovo variation, where you're only finding 60 or so variants per individual sample that you're looking for. So this is the easiest type of variation to find. What about structural variation? Well, if we compare different calling methods on the same data set, you tend to get this sort of flower of a Venn diagram of the different numbers and lack of overlap between calls that are made. And what this goes to highlight is the fact that there's a lot of uncertainty that's present in the type of calls that are made for structural variation using high-throughput sequencing. So why is that the case? Well, it's because by and large, variants that are larger than the length of sequences for high-throughput sequencing are detected in an indirect manner. So they're just inferred by either discordant distances between paired end reads or read depth methods. And so John gave a very good survey of this a couple of days ago. So only 20 to 30% of structural variant calls are detected with high throughput sequencing and so-called split read sequence analysis. And this is a type of method where you just simply observe the variant directly within the read itself. And this is the highest fidelity, but also, unfortunately, the lowest sensitivity for at least structural variation. So how can we do better? We can just simply apply longer reads. And this has come about through, in the last 5 years or so, they've gone from fairly inaccessible down to what's becoming essentially benchtop. Mike Schatz flew over here with his sequencer of methods to do single molecule sequencing, either Oxford Nanopore or Pacific Biosciences. They're starting to become fairly interchangeable. And we can see that if we compare 2 recent runs that were done, one of a Puerto Rican individual and another of, this is a Han Chinese, so the Puerto Rican individual, reads from the Puerto Rican individual are shown on the left, and reads from the Han Chinese from PacBio are shown on the right. What you can see is that, so by and large, they have the characteristics where the read lengths are cropped here at 1,000 base pairs, but they go from 1,000 in this case over 150,000, although the average read length is around 10,000 base pairs, actually in both samples. And there's also not a very big drop-off, or no real discernible drop-off of accuracy with read length. So what you can see is that there's this kind of large mass that is around, centered around 87 or so percent, and closer to about 90% in the nanopore sequences. So importantly, we could kind of define a cutoff, and we'd like to say, okay, what's the worst possible sequencing that we want to deal with? If we're coming up with methods that can handle, say, really noisy reads, how noisy do we care about? And so we could say a cutoff is, say, 85% and below is what you could consider fairly noisy. And so about 12.5% of this data set in the nanopore reads are below 85% identity, and about 30% of the PacBio reads are below this identity. So if we're detecting variation with these sequences, given alignments, it's actually extremely straightforward in many cases with appropriate alignments to detect structural variation. So what's shown here is a simple ALU insertion with reads that are aligned by Hung Lee's Minimap2 method, and this is a heterozygous event. And without any sophisticated methods, you could just simply scan through this and detect the variant. So unfortunately, this is something that for computational biologists, we don't necessarily like to see this too much, because some of the problems that are associated that we think are interesting to solve, such as detecting variation, are kind of going away in terms of technological development. But what we'll actually end up focusing on is solving harder problems, not necessarily more problems of this sort of easier type. Okay. So one question, which is some of the background about this type of study, is with an error rate up to about 15%, how do you efficiently align long reads? And it's not a very new question. In fact, this question's been around since for over 20 years, when gapped BLAST and BLAST-Z were created, for example. What's shown here are alignments, syntenic regions that are detected between human and mouse chromosomes. And so these are all the way up to 30% divergent regions, showing that it is very possible to do accurate alignment of very long but divergent sequences. The question is, how do we do it faster? So BLAST-Z was developed sort of in response to gapped BLAST being too slow. And then BLAST-Z, it turns out, is far too slow to work with the current data sets that we have, which involve gigabases of alignment that we'd like to do in a short amount of time. So if we're going to design an efficient single molecule sequencing read alignment program, it's probably going to be based on something very similar to what's been developed before, such as the BLAST or BLAST-Z or BLAST alignment. So what we'd like to do is find exact matches between the read and the reference, cluster these matches, and then refine them into pairwise alignments. So today I'll just talk about that first step. When talking about background, I'll talk about the first step of finding exact matches between the read and the reference. And so to start off with that, we have a question. This truncated some of the lettering. So there's 2 coin tosses. Unfortunately, I had a friend do this before, but I couldn't find his results, so I had to use one that I found on the web. One of these is created by a human, and the other is created by actually flipping a coin. And so the human just imagined a set of coin tosses, and then the other one is an actual set of coin tosses. And so by now, you've probably been able to recognize which one is the human-generated set of coin tosses and which one is the coin-generated set of coin tosses. So show of hands, how many people think that the first set of coin tosses is the set of tosses that are human-generated? We have 1. Not many. Okay. How about toss 2? Okay. So can anyone say why?
The first one has lots of straight H, like H, 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 H.
Yeah. So that's exactly right. So the first one has the longer run of heads, and it's very reassuring to see a room full of computational biologists appropriately applying their statistics. So unfortunately, the first time that I did this, I had to find a mechanical engineer to generate the set of coin tosses. And he didn't generate the long series of heads and tails. And so the first toss has 8 heads in a row, and the second one has 5. And so it turns out that you can check this. You can check for the number of runs with the Wad-Wolfowitz test, and you can show that the probability that the first toss was human-generated is very low, and the second toss is human-generated is very high. And you can compare this to the expected run of the longest run of heads or tails, which scales with the log of the number of coins that are flipped. So, okay, good. I don't have to go into too much detail explaining this. If the error rate is, say, 10%, we don't expect there to be one error every 10 bases all of the time. And since the errors tend to kind of have larger runs, we then expect there to be larger matches that we can anchor off of, even though the error rate itself is fairly high. And so back when we were first looking at this, we wanted to model this process, and we looked at something called the waiting length. And what this is the number of bases that are sequenced before sequencing a k-mer of a specific length that we're hoping to anchor off of with some probability. So what we can, with some error rate row, we can measure the probability that we see any word, 1 minus rho to the k, given some epsilon, which is the fraction that we're willing to, the certainty that we would like to have before seeing a base. We can compute this number of words that we're going to sequence before sequencing a word correctly of length k, and we can assign that to a value t. And then this, and the equation at the bottom is a solution for the number of bases. We're essentially solving for the number of words that are sequenced before sequencing a word of length k, such that all the previous words are smaller than k. And we can compute something called the waiting length. And we can use this to model the different waiting lengths across different accuracies of sequencing reads. And so what's shown here is a slightly different way of looking at this. We can look at the number of correctly sequenced k-mers per kilobase of sequencing read. And when I was first designing methods to map single molecule sequences, unfortunately the error rate was sort of hovering between 15 and 20%. And actually it was often greater than 20%. And so the hope of using matches of length 20 or greater with sequences that were essentially 1,000 bases was actually fairly hopeless at the time. But fortunately now, because the error rate is in this sort of range hovering between 10 and 15%, we're starting to see a fairly appreciable fraction of spans of fairly long length that contain correctly sequenced words. So that means that we can actually have fairly different design parameters between what we have used in the past for detecting matches between single molecule sequences and genomes, and what we're currently using to detect matches, exact matches. So the previous approaches were fairly reliant upon either Burrows-Wheeler transform or suffix array-based methods. And the basic idea is that you'd like to have some sort of tri-representation of the genome such that you can essentially try and thread sequences through this tri and find exact matches that way. And the reason for this is that you don't want to essentially have any fixed length word that you're hashing on, because you're not guaranteed with any particular sequence that it'll have any of those fixed length words to align. Not only that, but for the very short sequences, for the, say, 32 base pair reads that the very first Burrows-Wheeler aligner was based on, you'd like to do some sort of threading where with a 32 base read, you might not have any very long matches, and so you'd like to try different alternative sequences that can potentially match back to the reference. So let's look at that and try and see how the characteristics of that type of alignment compare with what we might want to use nowadays. So I'm showing this, the Burrows-Wheeler search. The suffix array search also has similar limitations, but so a Burrows-Wheeler transform. How many people here in their first algorithms class in their first year of grad school have gone over the Burrows-Wheeler transform? This is helping to design courses in the future. Okay, so unfortunately that takes about 15 to 20 minutes to go over, so we won't go over that now, but essentially what you have is you can take an original query string and you find all cyclical shifts, sort the cyclical shifts, and then create a column that represents the last character of the cyclical shifts. And then you have two extra tables that represent the frequencies of the nucleotides that occur at that base or before. And this all isn't super necessary to know for the time being. What's mostly critical to know is that for every base that you're matching, so we essentially match the first base of a read to all such, sorry, the Burrows-Wheeler transform allows you to do something akin to a longest common prefix or longest common suffix if you reverse a sequence. And so what you do is you essentially start at the end of a query and work your way back, expanding or limiting the search more and more per base that's added. For every base that you're searching, you're incurring essentially a count that's loaded from this count table that's roughly up to 1 billion entries long, although there's some sort of extra calculations that are done to compress that. And then some decoding of that. And so that actually takes a fair amount of time. And if we go and hack the BWA mem code, because you could kind of assume that's some of the most efficient code out there for doing this type of query, we can do about 141,000 queries per second using the Burrows-Wheeler transform. So that's pretty fast, but maybe we could do a bit better. So one potential way to do a bit better is to improve the cache locality. This is something that was done for RNA sequencing reads with a HiSat2 aligner by Dawei Kim. But that's not necessarily the approach that we'd like to have here. Maybe we could just have something that's strictly using a hash-based method to compare sequences. The reason being is that we have longer and essentially more accurate sequences, so we don't have to use this sort of try type of search in order to find alignments. So what's become kind of re-emerged as the rage is finding word matching using minimizers. So if you're going to be searching a read against a genome using fixed length sequences, there are essentially two options, or maybe three options, depending how you count things. But you can hash essentially all k-mers in the genome and compare all k-mers in the genome against all k-mers in the read. Or you can hash, say, a subset of k-mers in the genome, and unless you're using any specific sort of reduction function, you have to hash all k-mers in the read and compare that back against the genome. The reason being is that if you, say, were to try a strategy where you hashed 1 out of every 10 k-mers in the genome and exactly 1 out of every 10 k-mers in the read, there might not be an alignment between the correctly sequenced k-mers in the read and the correctly sequenced k-mers in the genome. So the alternative approach is you define a function that maps from some sequence to a reduced representation of that sequence. And that mapping that's fairly efficient and simple to use is called minimizers, where you essentially scan along a genome, and within some window, you emit the minimum value at that value and the position of that value. And that scans across the entire genome. So what's shown here is a simple example of a window size of 3 scanning across this integer valued sequence. And so what this does is it compresses this 12 integer set down to a 5 integer set. So we have a reduced representation of the sequence. And fortunately, anytime that you have a sequence that's at least of that window length that's also been, say, correctly sequenced in a read that you're comparing to the genome, then you're selecting the same minimizer between that read and the genome. So it's a way to go about mapping the two spaces between the read and the reference back to the same reduced representation. So why is this nice? Well, it turns out that it's a fairly compact data structure. So there's about 500 million entries if you use a window size of 10 and 17 MERS to do a search across the genome. And so previously, if you recall sorry, this keeps showing up. It's probably hard to see in the back because it cuts off the end, but the BWT search has about, it was 150,000 or so queries per second, and this is around 2 million queries per second. So the time that's required to search this index is considerably faster using the minimizer approach relative to the Burroughs-Wheeler approach, and you're able to, this is enabled by the fact that sequences are much longer and more accurate than they were when single molecule sequencing first started. So one of the things that you can look at is how often are you able to exactly align a read back to its correct position in the genome using something like minimizer-based mapping. And what's shown here is 4 different examples where what we have are a query of 1,000 bases, and we're looking for the number of essentially shared words between the query and the genome, and the number of other similar matches elsewhere in the genome, or the fraction of essentially unique calls within the genome. So what's shown here, it's a little complicated to read, but the basic gist is that moving sort of in a left to right, top to bottom fashion, what you're looking for is more shared matches between a read and a reference. So this is an example where you essentially have 1 k-mer that is being used to query a genome. And this is, say, 5 k-mers all the way up to 20 k-mers that are used to query a genome. And it's showing the amount of essentially unique matches. And so once you get up to 20 k-mers in a 1,000 kilobase region, you have essentially all unique matches between the read and the reference without even having to do any specific realignment. What does the minimizer structure look like for a real data set? So I spent the last few weeks writing kind of a test new aligner, and I wanted to at least have one slide that used it. And what this shows is the number of minimizer matches between reads and the reference for a simple PacBio data set. What you can see is that by and large, so the minimizers of course scale by the read length, and most reads have a very large number of matches between the reads and the reference. What this tells us is that it's getting much, much easier to do alignment now than before. We just essentially have to find minimizers and look for clusters of this order of magnitude between.
I can't see, unfortunately, my answer. Can you explain what is the x-axis and y-axis here?
On this guy?
Yeah.
So this is just the length of a read.
Okay.
It's just a scatter plot. And so I took a bunch of took reads, found the minimizers, scanned through, looked for essentially clusters, and I'm not getting into the details of how that clustering is done, but given the top ranked cluster between the read and the reference, how many minimizers are there?
The number of minimizers.
Yeah. And so, and it's about, I think it's around a tenth. The slope is about 0.1 here. I should have added the.
And K is towards Spartan.
K is 17.
So,
Yeah. Okay, so we've got plenty of time. Okay, so that part is just discussing methods for doing read mapping. And you kind of think of this, if you are going to be detecting structural variation using alignment, that's the first step that you're going to have to do. Nowadays, the Minimap aligner is so fast and pretty accurate, it's hard to really recommend doing anything else. So the method I wrote, Blazor, it's now about 10 years old. 10 years in code pro length is about the same as in dog years, so that program is pretty much ready to not be used anymore. I did some timing runs yesterday, and the current run was about to map about 100 million bases took 47 seconds with NGMLR and about 30 minutes with Blazor. So it's not terribly competitive anymore. It was good back in its day. Okay, so calling variation using SMS reads. So it's kind of a choose your own adventure type of study, where currently with most Illumina sequencing, there's sort of stock Illumina sequencing, where you go to New York Genome Center, you get back 30x coverage of 150 base pair paired end reads. With single molecule sequencing, it's still a bit of the Wild West, and we're kind of still deciding exactly what's the best approach to do any variation discovery. But there's sort of three different approaches that these sort of fall into, with two of them being almost identical. And so what you can kind of consider is, if your goal of your study is to detect structural variation without necessarily trying to find base pair resolution, and your input is somewhat low coverage, then the method that Fritz Sedzelek wrote, NGMLR plus sniffles, is a pretty good way of doing this type of detection of variation. If your study is to try and resolve local haplotype structure and to try and get base pair resolution of variation, the Denovo assembly program called Falcon Unzip is a pretty good approach to doing this. And then finally, if you would like to try and resolve essentially complete haplotype resolved information using high coverage single molecule sequencing and multiple data types that I'll get into later on, then there's a method that I recently wrote called phased SV. And so they kind of scale in terms of resource requirements from left to right on the screen, this being pretty straightforward to do with modest input, and this method requiring some pretty onerous data types and a fair amount of calculation. But what this does give you is essentially a very fine survey of the variants that are in a genome. Okay, so why do we need to take potentially different alignment approaches, and why does the NGM-LR approach work really well? So Mike Schatz showed a slide about this, was that a couple days ago? So here's a region where there's a 400 base pair tandem repeat expansion in reads that are mapped by Blazor. What you can see is that it's kind of scattered across this locus, and if you were using a fairly simple method to kind of go and scan and look for essentially reads that agree with each other at the same position, you're not going to find very many of such sequences. And furthermore, unfortunately, there's a bit of a bias here where red is the reverse strand and blue is the forward, or it's vice versa. But there's also a strand bias in where these gaps are placed. So that's clearly not the best approach to trying to find variation. The approach that Fritz took was to say, okay, well, let's actually try and design the gap parameter around finding variation. And so very importantly, the gap parameter gives essentially very low penalties to structural variant sized variants. And what this does is essentially pushes the gaps into fairly well defined regions. So these are images from the paper that describes NGMLR. There are plenty of regions that don't look this clean, but by and large, this is able to go about defining variants with very with pretty good sensitivity without having to resort to assembly-based methods. And so what that looks like is, since you're not having to assemble sequences, you can actually go and try and downsample and see what the ROC curve sort of looks like with lower and lower coverage. And what you can get is, with what should be fairly easily achievable, you can get, say, an 80% recall. So this is actually somewhat out of date. They've been pushing these methods further since the paper was published. And so you get 80 to 90% recall at about 10% sequencing coverage. So it's a fairly sensitive method to detect variation. Unfortunately, it only gives you the set of sequences, at least for the time being, that are that confirm a variant. It doesn't give you a very high accuracy consensus sequence. So to do that, it helps to find variation using de novo assembly. So unfortunately, for the time being, this also requires fairly onerous input requirements. So high coverage, about 40-fold single molecule sequencing, and it has a pretty high computational burden. So the first approach that was used for this was Smart SVU. This is now quite a few years old, and this was a local assembly-based approach where sequences were mapped back to the reference. Reads were collected in regions where there are expected to be structural variants, and they're also sort of collected in an unbiased manner, tiling across the genome. They're collected in windows of about 60 kilobases. Each window is assembled and then mapped back to the reference. And so the first thing that this did is it showed that there's a pretty large gain in sensitivity of variants relative to, say, the 1,000 Genomes project that relied on that relied on high throughput sequencing, where there are about 4,500 structural variants per allele in the 1,000 Genome study, and then there's 20 to 25,000 variants that are found per genome in these PacBio-based genomes. What's shown here are the number of essentially novel sites, novel variants in dark colors relative to the previous variation study. So it shows that most of the variants that are found in this first round of single molecule sequencing studies are novel with respect to new ones. So the question is, what type of variants are we finding in these? Turns out that most of it is simple tandem repeat and tandem repeat variation. So the first sort of pie chart of this showed that there was about 55% of the variation is tandem repeats. And then there was this big sort of chunk that was 17 to 20% or so that was unannotated. I've since kind of gone back and seen that most of these unannotated variants are actually just tandem repeats that Tandem Repeat Finder wasn't able to annotate. And so what you have is around 70% of the variants that are found in these data sets are tandem repeat variation. Unfortunately, that causes a problem because comparing different calls in tandem repeat regions is a pretty difficult task, and by and large, the gap penalties and essentially evolutionary models that we have for regions like tandem repeats don't necessarily apply, or sorry, the models that we have for something like mobile element insertion don't necessarily apply to the mechanisms that are being used to generate the evolution in tandem repeats. So it's an ongoing study to try and determine the best way to go about finding the alignment within tandem repeat regions. Ron Schmier actually just recently published a paper that has this edit distance that's based on duplications and deletions between sequences, and that's probably going to be the best way to define how. That was in the context of cancer studies, but you can also apply it in this particular type of case, but that's kind of ongoing research. So that was a local assembly-based approach. You can think about global assembly-based approaches. This is just an unbiased de novo assembly. While I was working on improving the local assembly methods, Jason Chin, who was an employee at Pacific Biosciences, wrote essentially a diploid assembler that does de novo assembly, but it also attempts to essentially phase and detect structural variants at the same time as doing de novo assembly. What's shown here is an assembly graph of a crossed Arabidopsis strain where there are two divergent strains that have structural variants between them, and every variant that structural variant between them corresponds to a bubble in this assembly graph. So running this Falcon unzip, this is the diploid assembler, on a Puerto Rican individual through that was sequenced in part of the 1,000 Genomes project, gave pretty similar results to what was done with essentially what the best approach that I could do with local assembly. So given about 40-fold PacBio sequencing of reads that averaged 10 kilobases, the main or the primary config N50 was about 16 megabases. The haplotype N50, so these are all of the extra sequences that could be resolved from the alternative haplotype, was about 200 kilobases. It doesn't really make too much sense to really try and define a haplotype N50 for these assemblies because they essentially only exist at the structurally divergent regions. And so you only have very small regions where you have haplotype differences. So total SV count, about 23,000 sites, 70% of which are heterozygous. And so what it shows is that it's able to in a very sensitive way detect heterozygous structural variation even when doing a full de novo assembly. But there is a bit of a word of caution that when doing de novo assembly, segmental duplication architecture is by and large not resolved. And so what's shown here is a plot of segmental duplications in the human genome where there's the length of the duplication on the x-axis and the identity of the duplication on the y-axis. And each dot here, each black dot corresponds to a segmental duplication that's been resolved within the de novo assembly. So by resolved, what I mean is that the assembly spans on both sides of the duplication. What this tells us is that we know the actual structure of the duplication in this region. If the assembly ends either before the duplication, which is shown in red here, or within the duplication, we call it unresolved. And so if you kind of change your gaze to the red dots, what you can see is that these are corresponding to the longer and more high identity duplications within the human genome. And so this plot looks essentially the same on every de novo assembly that we've applied this to, even with some of the most recent nanopore runs. So the runs that are getting the longer and longer read lengths, we're still seeing this sort of large cluster of long and highly identical segmental duplications that are not resolved. So there is actually a fair amount of functional variation that's within this. And within the 1,000 Genomes project, we looked at using read depth methods and compared the calls by read depth methods within regions of segmental duplication and saw that almost all of the calls that read depth methods were able to find were essentially within segmental duplications were missed by de novo assemblies, or essentially uncovered by de novo assemblies. I don't show it here, but this corresponded to about 314 genes within the entire data set that we're looking at. So there's a very massive functional consequence of this type of variation that's missed by current de novo assembly methods. And this is something kind of alluding to another talk that I'll give on Monday about methods to try and resolve these variants. And Faridoon's probably going to start coming up here soon. 5 minutes. So the last bit, I'll try and go over some of the work that the Human Genome Structural Variation Consortium, the HGSVC, has been doing to try and essentially comprehensively define structural variation in essentially 3 genomes, but 3 trios that we've been looking at. One from Puerto Rican, Yoruban, and Han Chinese. This is to try and span the range from admixed, highly divergent, and lower divergence within haplotypes. And essentially what we're doing is we're using every possible method that we can apply to these 3 genomes and to try and detect variation from them. And this types of methods scales from what I call short read, long read, all the way up to chromosomal scales. So short reads, we know what they are, Illumina sequencing. Long reads, we more or less know what they are, PacBio and nanopore. Also, we included some 10x genomics, although we only use this for phasing. And then the chromosome scale. And this is a kind of new approaches to providing very long-range information when doing phasing, and in the case of high C, scaffolding. So the type of technology most people are not familiar with is the strand-seq technology. What this does is it takes single cells, replicates them in the presence of BRDU, and does a single cell sequencing reaction that essentially is only able to sequence the strand that hasn't been replicated with BRDU. So every cell that's sequenced will have reads from only one or the other strand. So anytime that you see a read mapping to one strand on any part of the chromosome and the same strand elsewhere on the chromosome, those two reads are in phase. And so what this gives you is full chromosome to chromosome phasing of sequencing reads. Unfortunately, it's a somewhat low throughput technology, and so it's kind of being ramped up to being applied to more and more samples, but for the time being, high C, which gives you fairly similar results, is a bit easier to apply and higher throughput. So how this was applied to this data set is in this essentially a pipeline that we call phased SV, where we take strand-seq phasing, that's chromosomal phasing, which is fairly low coverage, so it's not very dense chromosomal phasing, overlay that on top of 10x phasing, which gives us very dense and chromosomal scale phasing. And we simply use that to partition single molecule sequencing reads by haplotype. And so now what we have is what was before a diploid assembly problem. We've been able to repartition the sequences according to haplotype, and now we've turned a diploid problem back into a haploid problem. So Tobias Marshall worked on the phasing part of this project, and we're looking at different ways to combine different technologies to do a large-scale phasing. What's shown here are phasing results using single technologies, and you can get all the way up to what's shown here is just the full chromosome scale that you can get with strand-seq and high C, all the way up to fairly large-scale phasing with 10x sequencing. What's shown here is different pairwise combinations. You can get very dense chromosome scale phasing with strand-seq. Also, what we can do is we can see the relative contribution to variation that's detected when trying to detect variation with both single molecule sequencing, Illumina, and say bio-nanogenomics. So what's shown here is a bar plot of structural variants in a Yoruban individual, and what's shown in red are variants only detected by PacBio, green only detected by Illumina, and orange both PacBio and Illumina, and so on for the 7 different combinations of either individual pairs or all agreeing technologies. And so the take-home message from this is that the largest contribution to the raw number of variants is relatively small-scaled variants that are detected by PacBio. We know that these are mostly tandem repeat variation. And as we move into larger and larger variants, essentially start to see more and more, a greater and greater fraction of essentially all methods starting to agree on the type of variants that are called. What's also nice with phased SV is we essentially get complete resolved haplotypes. So we have assemblies of the 2 haplotypes of all of the individuals. And what we can look at is interesting regions where you can, for example, find VNTR loci that are structurally divergent at the same locus across all haplotypes that are seen. And the Grim Reaper is heading over, so I'll wrap this up. I'll skip past some of the Illumina results and move on to some interesting results that we have based on the strand-seq data. So I mentioned that anytime you see a read on the same strand, it's in the same phase. If you see most reads on the same strand, but a swap of strands, what this corresponds to is a swap of the orientation of the DNA that's there, so you're finding an inverted sequence using this single cell sequencing approach. What it's able to do is define a very comprehensive set of inversion variants within even highly duplicated regions of the genome that, say, for example, can't be accessed by any of the other technologies, such as 10x technologies or bio-nanogenomics. This is work done by Ashley Sanders at EMBL, where looking at some of the inversions that we find within highly duplicated regions, there's this model where, depending on the duplication architecture that an individual has, you can be predisposed to microdeletion or duplications. depending on the inversion polymorphism that one has, you can be predisposed to microdeletions and duplications. And so having a full catalog of the inversions that are floating around in different haplotypes in the population can tell you what potential architectures are predisposed to new microdeletion duplication syndromes. And what Ashley is able to find in simply 6 different individuals, 25 new cases of inversions that are at the boundaries of potential critical regions. So this is a very massive leap forward in our understanding of genetic variation within highly complex regions that could be leading to disease or could be predisposing to disease. So thank you. There are many, many more people that have helped with all of this, but I'm kind of just listing the people that have been closest. So UCSD, Glenn Tessler and I worked on Blazor together. Washington, Evan Eichler, John Huddleston, Mitchell Volger has been working on some of the new assembly results, and then many of the members of the HGSVC. Any questions?
Time for a couple of questions.
Thanks, Mark. I was just wondering, so you talk about like combining multiple platforms. Is this like a way, basically what do you need from a single technology, like enough throughput or like the error rate, which will make it like just suitable just to detect like all the structural variants with no using like other technologies, like in that?
Sorry, could you phrase that again?
So you're combining different technologies, right? Like, but I'm saying is like, can you stick with a single technology? And if so, which properties of that technology do you need? Can you just if combine increases the throughput?
Yeah.
The coverage, will you be okay with that? Or you still need like good, like having no errors?
Yeah, so the main way that technologies were combined in the HGSVC was essentially to do phasing and then variant calling. And so what this allowed was, for all of the regions where there were pretty small variants in the PacBio data, where it would have been pretty difficult to kind of tease out the different haplotype structures, by phasing and partitioning, you're able to have better resolution of that. We didn't really use the, say, Illumina SV calls, combine them with the PacBio SV calls. We just kind of made a merged set altogether. Essentially, one of the things that one could do is use the Illumina calls to inform whether or not maybe the methods aren't working quite as well in the kind of emerging PacBio approaches, or sorry, SMS approaches. By and large, you can show that essentially any variant that you can find with Illumina sequences, there's more than enough information content in single molecule sequences to find those variants as well.
When you're comparing the aligners, did you consider error-corrected data, or if you didn't, what's the effect of error correction on the performance of aligners?
So no, I didn't do that. Well, the error correction problem is an alignment problem in itself. And so because of that, the easier problem is reads back to the reference. And then it's a little bit harder to then go and try and do, say, so the problem of say, mapping Illumina sequences back to a single molecule sequence at the alignment phase instead of, say, the assembly step, there's not a lot of information within, you could sort of consider the 125 base pair region in the single molecule sequence as opposed to the Illumina sequence. And it's sort of the same problem between mapping that 125 base pair SMS sequence back, and then that's actually a pretty difficult alignment problem. Alignment only gets really easy with the amount of information that's contained in a few thousand bases or more of a low identity or low accuracy sequence.
I'm wondering, we published this method called color map for correcting PacBio reads with Illumina reads in a blind way. I mean, we never really thought whether it makes sense to do that and then call structural variants, or with the integrated analysis through comparing the structural variation calls makes sense. Is there a principal way to think about what is the right approach?
So I think, so it makes sense until you get to some of the really difficult regions where the mapping starts to get very ambiguous. So it's definitely a principled approach for much of the genome, but then it becomes somewhat difficult in other parts of the genome. And I know in my mind, it seems an easier problem to look at some sort of consensus between the single molecule sequencing reads first, and then maybe look for additional support of Illumina sequences later. But people have definitely shown an improvement over simple single molecule sequences. So
There should be a mathematical distinction. I can't.
yeah, I mean, like I said, you could look at just simply the information content that's involved in 150 bases of a 85% accurate sequence and just show that that information is relatively low compared to, I mean, it's just the scales of the length of the sequence. So, but the key is that it becomes very difficult to solve that mapping problem in duplicated regions, which tend to be the regions that have the variance.
Especially tandem ones. I mean, say same finger proteins kind of a scenario is easier, or VDJ sequencing is a little easier on the genome, the germline, just because there's a gap between successive V segments, and you can correct the errors in between pretty accurately, and then PacBio becomes nothing. As long as there's a little hook, little anchor that you can correct pretty accurately, you can do something like that.
Right, yeah. So it's a little, sometimes can be misleading to have summary statistics on all sequences. We probably want to take this offline, so.
Okay.
All right, thanks guys.